Hey everyone, Dr. D here, and in this video we are carrying on with our Talaros Foundations in Microbiology 12th edition by Chess textbook, chapter 6. Let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, welcome back. Chapter 6. It's all about viruses, viroids, and prions. These are known as small infectious agents. These are not living organisms. So let's start with viruses. Viruses are obligate intracellular parasites. Remember what that means. Anytime you see the term obligate, that means it is a must, it's a requirement. So these things are required to parasitize the inside of the cell. They need to get inside of bacteria or inside of protists, inside of fungi, plants, and animals. Basically, viruses need to get inside of host cells, living cells, in order to actually reproduce and propagate. They are ultra microscopic in size. Do you remember what ultra microscopic means? It means too small to even see with our light microscope. They are smaller than even the smallest bacteria. They range from 20 nanometers. Nanometers are tiny. Remember, a nanometer is 10 to the minus 9 meters. So, I mean, these things are super tiny. They range from 20 nanometers all the way up to 750 nanometers. So again, when we're dealing in the nanometer range, that's the range of an electron microscope. Remember, a microscope is good for organisms that are at least 1 micrometer or more, okay? An electron microscope is good for anything smaller than a micrometer. And again, you remember, these are not cellular in nature. So they are small infectious agents. We do not call viruses organisms. We do not call them living. Instead, we call them agents, and we call them active or inactive. And one of the main reasons they're not living is, well, they're not made up of cells, but also they do not independently fulfill the characteristics of life. Remember all those characteristics of life that we touched on in chapter 1. So basically, these viruses do nothing when they're outside of host cells. They're inactive macromolecules outside of host cells. When they're outside of host cells, all they can do is hope they bump into a new host cell, really. That's really all they can do. Once they bump into a new host cell, that's when they become active, once they're inside of a new host cell. Every virus at its heart has two main components, okay? Every virus has to have these two main components. A protein shell called the capsid and a nucleic acid inside. And do you guys remember that the nucleic acids are DNA or RNA, right? RNA counts as nucleic acids as well. I want you to see something real quick. Nucleic acid of a viral genome is either DNA or RNA, but not both. What does that mean? It's really interesting because if I were to ask you about your genome, you would say your genome is made up of DNA, right? And some viruses have a genome of DNA. However, did you know that some viruses, they don't have a genome of DNA at all? Their genome is made of RNA. Isn't that neat? So that's strange. Living things do not have genomes of RNA, but viruses can have genomes of RNA. That's why it says surrounding nucleic acid, because some viruses use DNA for their genomes. Other viruses use RNA for their genomes. But it's not both, right? No virus has both DNA and RNA genomes. It's either a DNA virus or it's known as an RNA virus. But either way, your genome surrounded by a protein coat called the capsid, together it's called the nucleocapsid. So that's why I say every virus at its core has a nucleocapsid, because you have the nucleic acid, either DNA or RNA, and a capsid, a protein coat around that. And it gets stranger, like really strange. The nucleic acid can be double-strand DNA for some viruses, Other viruses might have single-stranded DNA. Some viruses have single-stranded RNA, and some viruses have double-stranded RNA. So they have quite the diversity in the types of genomes that they can have, whereas living things have to have double-strand DNA for their genomes. Additionally, viruses need molecules on the surface of the virus in order to specifically bind to host cells. These are normally called spike proteins, but we'll get into that in a minute. Now, once a virus is inside of a host cell, it basically takes over the host cell and makes it into a viral factory, like a little machine factory. Viruses multiply by taking control of the host's genetic material and regulating the synthesis and assembly of new viruses. So for instance, they'll take over your ribosomes. And remember, your ribosomes should make your proteins, right? Well, guess what? They take over and then they're making their own proteins, their own capsid proteins, their own spike proteins, their own proteins with your ribosomes. So they basically hijacked your cell and turned it into a little virus factory. Viruses lack enzymes for most metabolic processes. Viruses lack machinery for synthesizing proteins. So how are they making all these things? They're making these things by using your cellular machinery. Your cells become factories for making more viruses. Viruses can't make more of themselves. And do you remember when I told you that viruses are ultra microscopic? Anything smaller than about 1 micrometer, you're not really going to be able to see it with a light microscope. And most viruses are smaller than 0.2 micrometers. So you're really not going to see them with our light microscope, right? Instead, what do we require? Anytime you see a picture of a virus in a textbook, you should know that it was most likely captured by an electron microscope. Because do you guys remember that E. coli is about 2 micrometers long? Streptococcus is about 1 micrometer wide. So you can see bacteria with a light microscope. However, once you start getting into the realm of the viruses, look at mimivirus here at 500 nanometers, which is 0.5 micrometers, okay? And then you have, and that's one of the bigger viruses. This is one of the bigger viruses. And then pox virus at 400 nanometers, herpes virus at 150 nanometers. Look how small these are. Rabies virus, HIV, influenza, adenovirus, T2 bacteriophage, polio, myelitis, and hemoglobin. This is a hemoglobin protein. By the way, look at this red circle. This red circle represents one of your red blood cells. So imagine your red blood cells are some of the smallest cells in your body. And look how tiny these viruses are compared to your red blood cell, right? So again, do you guys remember eukaryotic cells are the largest, followed by prokaryotic cells, and then followed by the acellular viruses, those small infectious agents, which are ultra microscopic and require electron microscopy. And remember that viruses bear no resemblance to cells. They completely lack protein synthesizing machinery. You know what they mean by protein synthesizing machinery? Can you beat Wicket? That's right, Wicket. They mean the ribosomes, right? They don't have ribosomes. They don't have ribosomes at all. They're probably the same size as a ribosome. So here you have viruses contain only the parts needed to invade and control a host cell. So essentially, what do you have? The virus particle has a covering called the capsid. Remember, a protein capsid. All viruses have a protein capsid. Think of it as a protein coat. Some viruses have an envelope, so essentially a membrane surrounding the capsid. And then viruses have a central core. Remember, inside of the capsid, you have your nucleic acid genome. And do you remember, viruses can either have a DNA genome or an RNA genome, but never both. Some viruses also have matrix proteins and certain enzymes as well, but not all viruses. So again, all viruses have capsid protein coats, which enclose the nucleic acid. The capsid together with the nucleic acid is called the nucleocapsid. Remember, all viruses, at the core of every single virus is a nucleocapsid, the capsid coat plus the genome. And then in addition, sometimes some viruses, some species of viruses have an external covering called the envelope. Those viruses that lack an envelope are called naked viruses. So some viruses are known for having an envelope, like HIV, and some viruses are known for not having an envelope, like a bacteriophage. And by the way, the capsid protein coat is not made of one big protein. It's actually made up of many identical protein subunits that link together. And these many identical protein subunits are called capsomeres. So you can think of it as capsomeres are the identical building blocks that assemble to form a capsid. So here you're looking at what's at the heart of every virus. Look at this. This is the capsid. Look, this is the protein coat called the capsid. And then inside you have some nucleic acid. And like I said, that could be DNA or RNA. And remember, together, the capsid and the nucleic acid are called the nucleocapsid, right? And here, so this one here is called a naked virus. It's called a naked virus because look, outside of the capsid is no envelope. However, look at here. Here, you actually have an envelope. See in purple? Here you have an envelope surrounding the nucleocapsid. The capsid is in green. The nucleic acid is in orange inside. And then look, outside you have the envelope, and this is known as an enveloped virus. And by the way, look, on the surface of the virus are these proteins called spike proteins. And you can find spike proteins on both enveloped viruses as well as naked viruses, even though they didn't draw it here. Spike proteins, their job is to bind specifically to the host cells that they recognize. Okay, so spike proteins are vital for binding to the host cell. And by the way, do you see how the shape of this capsid, this shape looks like a 20-sided dice. You know, if you have ever played role-playing games like Dungeons and Dragons and you have to use different types of die, you know, like a 4-sided die or a 6-sided die or, you know, 20-sided die. This reminds me of a 20-sided die. That's known as an icosahedral shape. It's a 3-dimensional symmetrical polygon with 20 sides and 12 evenly spaced corners. That's known as an icosahedral capsid. That's one of the 2 capsid types that's very typical. The other type of capsid is called a helical capsid. This is a continuous helix of capsomeres forming a cylindrical nucleocapsid. So when you think of capsids, when you think of protein coats for viruses, you should think they're either helical or they are icosahedral, right? Those are the 2 main types. Those are the 2 most common capsid types. Here's an example of a helical capsid. This is Ebola virus at the bottom here. A rod-shaped capsomere assembles into hollow discs. The nucleic acid is inserted in the center of that disc. And then you have elongation of the nucleocapsid from both ends as the nucleic acid is coiled inside. So what you have here, this would be Ebola virus. And see how it forms a long structure, right? That's a helical capsid. Inside of that helical capsid would be the DNA or the RNA genome. Whereas icosahedral capsids, remember, icosahedral capsids form this 20-sided dice structure, and these capsomeres come together to form these different facets that all assemble to form this 20-sided dice structure. And remember, spike proteins can poke out from the capsid itself. And by the way, icosahedral viruses may or may not have an outer envelope. The same thing with helical viruses, by the way. Helical viruses may or may not have an outer envelope too. But here you can see a naked icosahedral virus. This is called rotavirus. And here it is again as a cartoon image. So this is a computer rendering of rotavirus. This is an actual electron micrograph of rotavirus. And notice how it doesn't have an envelope. It simply has its icosahedral capsid, and then it has the spike proteins. The spike proteins are extending out from the icosahedral shape, right? Capsid. These little spike proteins, remember, they are responsible for attaching specifically to receptors on the surface of host cells. Here you can see, on the other hand, enveloped icosahedral virus. This is herpes simplex virus. It's an enveloped virus. And notice that here you have your DNA core, then you have your icosahedral capsid in blue. And what do we have outside of that in green? We have the actual envelope, right? So this is known as an enveloped icosahedral virus, whereas rotavirus is a naked icosahedral virus. By the way, when it comes to an envelope, it's usually animal viruses that are enveloped. And that's because animal viruses acquire the envelope when the virus leaves the host cell. They don't actually synthesize their own envelope. They kind of steal the envelope from the host cell. Isn't that neat? So here you can see, this is actually really neat here at the bottom. This is a cartoon of flu virus. And flu virus actually has, if you look closely here, numerous helical capsids. See, look, numerous helical capsids inside of an envelope, And so that is known as an enveloped helical virus. And I want to show you these spike proteins, hemagglutinin spike and neuraminidase spike, right? These spike proteins are essential for attachment of the virus and entry into the host cell. Now you might be wondering, what is the function of the capsid or envelope? Well, essentially it protects the nucleic acid when the virus is outside of the host cell. It also contains those spike proteins which assist in binding to the cell surface of the host, because viruses are very specific in the hosts that they bind to. In fact, they bind to specific receptors on specific host cells. There's also a third category for the structure of viruses. Remember, there was the helical viruses, the icosahedral viruses, but there are also what are known as complex viruses. And these are different because they don't have one particular shape, right? So they are complex. These are also known as atypical viruses. So an example is pox virus, or the different pox viruses which lack typical capsid, and they are covered by a dense layer of lipoproteins and coarse fibrils. Additionally, there are these bacteriophages that have kind of a combination of a polyhedral or icosahedral head along with a helical tail and attachment fibers. So let me show you what I'm talking about. Here you can see vaccinovirus, which is one of the pox viruses on the left. Notice that it doesn't look like a cookie cutter shape. In fact, you have the nucleic acid in the center. Then you have a membrane, a core membrane, which is really interesting. Then you have what is known as a lateral body. You have soluble protein antigens. You have an outer membrane as well, or an outer envelope. So you see how this is a complex shape here with the pox virus. And on the right, you can see this is a bacteriophage. It's a naked virus. And notice how it's complex because it has the icosahedral head, and then it has a helical tail, and then it has these tail fibers along with tail pins. These are known as complex viruses because they have that complex shape. This leads to our first concept check. How would you describe this virus? If you look to the right, that reminds me, that looks like the flu virus. Okay. And do you remember the flu virus? I told you that it has numerous helical nucleocapsids inside of an envelope. So the answer should be helical and enveloped. D. Now remember, a viral genome can either be DNA or RNA, but never both. That genome carries the genes that the virus needs in order to invade the host cell and redirect the cell's activity in order to make new viruses. That's essentially what the genes are coding for, are, spike proteins, capsid proteins, enzymes, anything the virus needs in order to invade and replicate itself. And the number of genes varies for each virus. Some viruses only have a few genes. Some viruses can have thousands of genes, but typically viruses have far fewer genes than life, right? Than living cells. And remember, there's so much diversity with regard to the nucleic acids of a virus. DNA viruses can have double-stranded genomes. Those are shortened to DS genomes. But they can also be single-stranded genomes or SS genomes. They can be RNA viruses. RNA viruses are usually single-stranded RNA, but it can also be double-stranded RNA. They can even be segmented into several RNA pieces or separate RNA pieces. So for instance, do you remember I showed you the flu virus? The flu virus is an RNA virus, but it's segmented into several pieces of RNA. So kind of like you and I have 46 chromosomes, the flu virus has numerous pieces of RNA as its genome. Isn't that interesting? Single-stranded RNA genomes that are ready for immediate translation. That means immediately to be read by ribosomes to produce proteins are called positive sense RNA genomes, whereas single-strand RNA genomes that must first be converted to proper or positive sense are called negative sense RNA genomes. I'm going to explain this in more detail in a little bit, but essentially what it means is there are two types of single-strand RNA genomes. There are two types. Positive sense single-strand RNA genomes and negative sense single-strand RNA genomes. What you should write down in your notes is that positive sense single-strand RNA is ready to be translated by ribosomes because it's similar to mRNA. Okay, write that down. However, negative sense RNA genomes are not ready for immediate translation by ribosomes because they are the complement to mRNA. They are not recognized by ribosomes. So viruses that have negative sense single-strand RNA genomes, unfortunately, they first have to convert their genome to positive sense, which is the complement to their negative sense genome, and only then can the ribosomes actually make sense of the information on the genome. I can explain this in more detail in a little bit. All right, so here's what I've been talking about with positive sense single-strand RNA versus negative sense single-strand RNA. Remember, positive sense single-strand RNA genomes are ready to protein synthesize. So that means that the ribosomes can attach directly to this genome and treat it like mRNA in order to read and translate those genes into viral proteins. However, look at this. Negative sense single-strand RNA genomes, the ribosome has no idea what this information is saying. So in this case, viral enzymes have to convert this negative single-strand RNA to positive single-strand RNA, which is the complement. The complement of negative is positive. And now the ribosomes say, oh, I know what to do with this. And they start translating these genes to proteins. I hope that makes sense. And if you have double-strand RNA, then guess what? What's cool about a virus having double-strand RNA is that one of the strands is the positive sense and the other strand is the negative sense. So the ribosomes will simply attach to the positive sense and translate that into proteins. So essentially, I already told you about some proteins that are necessary for viruses, right? Remember the capsomere proteins that assemble to form the capsid? Remember the spike proteins that help with adherence to the host cell? But some viruses also have polymerases. These are enzymes that can either synthesize DNA or RNA. Some viruses have replicases, which copy RNA, and some even have what's known as reverse transcriptase enzyme, which can synthesize DNA from RNA. And notably, this is the HIV virus, or retroviruses are known for having this reverse transcriptase enzyme. Now, how do we categorize and classify viruses? There's actually a committee, the International Committee on Taxonomy of Viruses, that lists 59 orders, 189 families, and 2,224 genera of viruses. With the nomenclature for families, families are italicized and given the suffix viridae, and genre are likewise italicized and end in virus. By the way, there's a new way of classifying viruses known as the David Baltimore method, and that's more prevalent nowadays. This is more reminiscent of how taxonomy works with living organisms. However, you guys know that viruses are not alive, so trying to classify them into families and classes and orders and genre is kind of silly. Nowadays, we classify them based on the genome they have. So if they have, for instance, if they have double-strand DNA, they would be in one category under the Baltimore system. If they have single-strand DNA, they would be in another category under the Baltimore system. So here you can see some important human viruses, and these are all DNA viruses. For example, let's look at the nucleic acid type. This is herpes viridae, herpes virus. The family would be herpes viridae. The genus simplex virus. Here, the common name is herpes simplex virus 1 or HSV1. And here's the name of the disease that it causes: fever, blisters, and cold sores. So again, notice how the family ends in viridae, whereas the genus ends in virus. And then there's usually also a common name as well. Here are some common DNA viruses, and notice how they range in size, shape. Some are naked, some are enveloped, some are complex. Okay, and then we have the RNA viruses as well. Notice how they have a wide range of shapes, sizes, envelopes, etc. as well. All right, now before we hop into how these viruses multiply, how they replicate themselves, let's take a quick break time with Gizmo and Wicket and see what these little guys are up to, and come back strong to finish off this chapter. All right, welcome back from break time with Gizmo and Wicket. It's always fun to see what these little guys are up to. Let's carry on with modes of viral multiplication. So in general, you have these phases in animal virus multiplication. The first step is called adsorption, followed by penetration, uncoating, synthesis, assembly, and release. These are the steps involved in viral replication. And by the way, adsorption is the most important step. This is where the virus binds to specific molecules on the host cell. And remember, it does so with the help of spike proteins. So let's talk about adsorption first. Remember, step 1 is called adsorption, not absorption, adsorption, which means to attach. The virus attaches to its host cell by specific binding of its spikes to cell receptors. See, look at this. This is a virus. And look at this. The surface of the virus has these spike proteins, and these spike proteins bind specifically to receptors on the surface of the host cell on the left. This is a very specific binding. This is not some random binding. So this means that different viruses have different host ranges, depending on what cells possess the receptors that are recognized by the viral spike proteins. So for instance, have you ever heard of the virus, hepatitis B virus? This virus only infects human liver cells. Why? Because only human liver cells, or hepatocytes, possess those particular receptors. The rest of the cells in your body, they don't have the receptors that are recognized and that bind to the spike proteins on hepatitis B virus. Whereas poliovirus is a primate intestinal and nerve cell specific virus. So any primate can be infected by poliovirus along its intestinal and nerve cells. Rabies virus, on the other hand, oh wow, rabies virus can infect many different organisms. And that's because it recognizes receptors on various cells, not just one cell type or two cell types, but various different cell types of many mammals. This is why, you could catch rabies from a chipmunk bite, right? And then, or a dog bite, or a bat bite, right? So there are many different animals that are infected by the rabies virus. However, hepatitis B virus has a much more narrow host range, having only the receptors on the human liver cells. Here you can see exactly what I'm talking about. Look here. The specific spike protein on the surface of this red virus is binding to a very specific receptor on this yellow host cell at the bottom here. And you can see another example right here where this naked virus has spike proteins in green, and they are binding to very specific receptors on the surface of this other host membrane as well. So one thing you should take away is that adsorption is very important. It is a very specific binding between spike proteins and receptors on the host cell. Let me put it this way. If a virus can't bind to your cell, then it can't infect that cell. Okay? So it's very important that the virus be able to bind specifically to some protein or structure on the surface of your cells in order to infect your cells. If it can't adsorb, it can't infect. And remember, the next step is penetration followed by uncoating. During penetration, the animal virus must penetrate the cell membrane. That means get inside, right? It needs to get on the inside of the host cell to deliver the viral nucleic acid, either DNA or RNA, into the interior. And how is penetration done? Penetration is done, most viruses enter through one of two ways, either fusion or endocytosis. So let me show you both. The top example here is entry of the virus or penetration of the virus through fusion. So let me show you how that works. Let's go step by step. The first thing that happens is that the virus binds with its spike proteins to specific receptors on the surface of the host cell. That was called adsorption, right? Attachment. Then there's a fusion. What does that mean? Membrane fusion. The envelope of the virion, virion is singular for virus, fuses with the cytoplasmic membrane. So the envelope of the virus fuses with the envelope of the host membrane. And then what happens? Because of that fusion, the viral envelope actually becomes one with the plasma membrane, with the cell membrane, and that allows for the viral envelope remains part of the cytoplasmic membrane, and so it basically just releases the nucleocapsid inside. You see, so now the nucleocapsid is now released into the cytoplasm. Isn't that neat? So that's called fusion. Now let me explain endocytosis. Again, remember we have to have attachment or adsorption. Specific spike proteins on the surface of the virus bind to specific receptors on the surface of the host cell. And then here's what endocytosis, remember endocytosis is called phagocytosis, where the membrane wraps around, the host cell membrane wraps around, you see, wraps around the virus and then brings that virus in a vesicle, right? So the envelope of the virion fuses with the endosomal membrane. And then after that, you'll have your uncoating, where now you can release the actual nucleocapsid later. So again, you can either have fusion or endocytosis. Those are the two main ways by which the virus penetrates the host cell. And then do you remember what comes next? Uncoating. Uncoating. Let's talk about uncoating. So look, once the nucleocapsid is inside, we need to get that DNA or that RNA, essentially that nucleic acid, we need to get it out of the capsid coat, because that nucleic acid is what's going to take over the cell. So we need to uncoat the nucleocapsid. That means break apart or disassemble the capsid coat. The capsid coat disassembles, releasing the nucleic acid, be it DNA or RNA, into the cell. And that nucleic acid is what takes over the cell. So you see, that's called uncoating. Here you can see uncoating again. See, when the nucleocapsid is freed from this vesicle, the nucleocapsid needs to break apart. The capsid part needs to uncoat, right? Uncoat. And what are you uncoating? You're uncoating the nucleic acid. And that nucleic acid is what actually takes over the cell and makes it into a virus-making factory. Next, what we need to do is synthesize, right? We need to make more viruses. That's called the synthesis step. We want to make more virion. We want to make more viruses. Now, what's really neat is that they've discovered that DNA genome viruses generally replicate and assemble in the nucleus, whereas RNA genome viruses generally replicate and assemble in the cytoplasm. That might be something to remember. And do you guys remember that positive sense single-strand RNA contains the message for translation? That means it can be directly translated by ribosomes, whereas negative sense single-strand RNA viruses, they can't be directly translated by ribosomes. First, we must synthesize the positive sense RNA, and then the ribosome knows how to make those proteins. Now, we've created all the parts that we need, right? We've created more nucleic acid, we've created more capsid proteins, synthesized the spike proteins, synthesized any enzymes that are needed for the virus. Now it's time to put them together like Legos, right? Put the virus together. That's called assembly. So this means that mature virus particles are constructed from the growing pool of parts, right? So we're putting these parts together. And guess what? Once you've put all those parts together, the virus is now ready to leave the cell. That's a process known as release. And why would you want to leave the cell? Well, because you want to go ahead and infect the next cell, right? So the virus is ready to leave the host cell. And this also happens usually in one of two ways. Budding or exocytosis, this results in enveloped viruses, or lysis, which is like rupturing the cell. That results in non-enveloped or complex viruses. These are the two main ways by which virion escape or leave the cell. Here you can see budding in process, right? This is called budding or exocytosis. Let's take a look at this. So here we have the nucleocapsid here in red and green, the nucleocapsid. And look what it does. It comes up and touches the plasma membrane or the cell membrane of the host. And in fact, what's really neat is it's pre-implanted viral spike proteins into that membrane. So the virus already put spike proteins, viral spike proteins in this envelope, in this membrane. The nucleocapsid touches the membrane, and then look, this exocytosis occurs. This is also known as budding, okay? Make a note of that. This is called budding. It's an exocytosis. Notice how the nucleocapsid is leaving the cell, and as it leaves the cell, it takes that envelope with it. The envelope was part of the cell membrane of the host cell, and these little spike proteins were pre-implanted in that membrane. Isn't that neat? And then the nucleocapsid extrudes from the host cell, becoming coated with matrix proteins and envelope with protein spikes, right? The protein spikes. And then the new virus is released, and now it's called an enveloped virus. So if I ask you this on the exam, let's see if you can beat Wicket. True or false? Viruses produce their own envelopes. That's right, Wicket. That's false. They don't produce their own envelopes. They stole the envelope. They stole that membrane from the previous host, right? So false. That answer would be false. Isn't that neat? And so if you ever wondered, like, how come some viruses have an envelope and some viruses don't have an envelope? Well, the ones that do have an envelope likely left the previous host cell through budding. And when you bud out, you're going to take with you part of the host's membrane, and that becomes your envelope. Isn't that neat? By the way, this is really neat. This is an actual picture with an electron microscope of HIV virus in red budding from the surface of an infected cell in blue. This is the infected cell in blue. This is the virus budding. You see how it does exocytosis or budding? And now it's taken the envelope from that cell. So yes, HIV is known as an enveloped virus. And now you know where that envelope came from. It came from the cell that it had exited from via budding. And by the way, you might be wondering, well, then how come some viruses are naked, right? The naked viruses usually lyse or rupture the cell, right? So imagine, okay, look at this. Imagine if the virus caused the cell to just break open, right? It just caused the cell to lyse or break open and pop open. Wouldn't that mean that all these little nucleocapsids pour out without ever getting an envelope, right? So that would be an enveloped, that would be a naked virus, right? So naked virion likely lysed the previous host cell, whereas enveloped virion likely budded or, exocytosed from the previous host cell. I hope that makes sense. And that leads us to our third concept check. Viruses commonly contain both DNA and RNA. Let's see if you can beat Wicket. Do viruses contain both DNA and RNA genomes? No, that's right. It's DNA or RNA genomes. So the answer should be false. Perfect. And by the way, animal viruses, like the viruses that infect your cells, they can have damaging effects on your cells. These are known as cytopathic effects. This is cell damage that alters the microscopic appearance of your cells. Your cells, if your cells have been infected by viruses, those cells could look disoriented under the microscope. There can be gross changes in the shape or the size of your cells. And there could be intracellular, that means inside of the cell, changes in your cells as well. Such changes include inclusion bodies or syncytium. Let me explain both. Here you can see this cell in the center. You see, doesn't this cell in the center look different from the other cells surrounding it? Well, that's because this cell in the center has formed a syncytium. A syncytium is when multiple cells fuse together. So because of the viral infection, two cells have fused together to form you can see two nuclei in the same cell. And that's because the two cells have formed a giant cell. And that's something that's common among virally infected cells, the formation of a syncytium. two cells fusing together. And then look on the right. This is an example of another cytopathic effect. This one shows cells that are infected with the varicella zoster virus. That's the chickenpox virus. And it's showing signs of inclusion bodies. See these punctate marks here? That the punctate means these little dark spots. Those dark areas show masses of viruses or damaged organelles. So again, virally infected cells show cytopathic effects, including inclusion bodies and syncytium, disorientation, and gross changes in size and shape. And some viral infections are persistent and some are acute. Acute viral infections come on strong, but then leave pretty quickly. Persistent infections last for an extended period of time. They're also known as chronic infections. The cell harbors the virus and is not immediately lysed. This can last weeks or the host's entire lifetime. Several can periodically reactivate. That's called the chronic latent state. So I'll give you a couple examples here. Herpes simplex virus, 1 and 2, which cause cold sores and genital herpes. These can reactivate time and time again. They go dormant and then they come back and reactivate, right? That's a chronic latent state. The herpes zoster virus that I just showed you, the one that causes chickenpox and shingles, this one can also go dormant for years upon years and years and then reactivate as shingles as well. And unfortunately, several strains of viruses can cause cancer as well, and that's a form of viral damage. Some animal viruses enter the host cell and permanently alter its genetic material, resulting in cancer. These viruses are termed oncogenic. Viruses cause cancer. And their effect on the cell is called transformation. When a cell goes from a normal functional cell to a cancer cell, it's called a transformed cell. Transformed cells have an increased growth rate, alterations in their chromosomes, and the capacity to divide for indefinite time periods. This means that they become immortal, and that results in tumors. Mammalian viruses capable of initiating tumors are called oncoviruses. And here's some examples. Certain strains of the papillomavirus, you've heard of HPV, human papillomavirus. Those strains of papillomavirus can cause cervical cancer. And certain strains of the Epstein-Barr virus, the same virus that gives the disease mononucleosis, this can also result in a type of cancer known as Burkitt's lymphoma. So now that we've discussed animal viruses, let's turn our attention to bacterial viruses. These are known as bacteriophage. When you see this term bacteriophage, you should think of virus that infects bacteria. These are also known as phages. The most widely studied of the phages is the one that infects E. coli. Viral multiplication is very similar to that of animal viruses. The only main difference is that the nucleic acid is usually DNA, and that DNA enters the cytoplasm directly, making uncoating not necessary. I'm going to show you how that works in a second. And release is a result of cell lysis by the viral enzymes and accumulation of virus. This is called the Lytic cycle. Let me ask you this. Let's see if you've been paying attention. If release is a result of cell lysis, do you think these bacteriophage are enveloped or naked? Can you beat Wicket? That's right, Wicket as always. These are naked viruses, right? Because if the cell just lyses open and all the viruses pour out, there's no budding that occurred, and so these are going to be naked virion. Very good. But again, notice that the steps in phage replication of a bacteriophage are very similar to that of an animal virus, except uncoating is not necessary. See, you have adsorption, penetration, replication, assembly, maturation, and release, okay? We don't need uncoating, and I'll show you why in a minute. And what's really neat about these bacteriophage is that occasionally the virus enters a reversible state of lysogeny, and its DNA is incorporated into the host's genetic material. So I'm going to show you what that means in a second as well. But essentially, bacteriophage, several of them, the ones that are known as temperate phage, they can either enter a lytic cycle, where they just get to business and explode the cell, right? Or they can go into what's known as lysogeny, where they lay low and integrate their genome into the host genome. It's kind of neat. It's almost like they have two options once they get in. They can either get busy, destroy the cell, and get out, or they can lay low for a while, right? So I'm going to show you how lysogeny works for temperate phages. All right, here's the multiplication cycle of the bacteriophage. In this case, it's one of the T-even phages that infect E. coli, one of the best studied phage. So take a look here. Let's look at step 1. Remember what step 1 is? Step 1 is called adsorption. Remember, this is where the bacteriophage attaches with its spike proteins to specific receptors on E. coli. Okay, so this is the E. coli rod-shaped cell. It's a bacillus-shaped cell. This circle represents its nucleoid. Remember, it's single circular chromosome, and this is the bacteriophage. And let's look at step 2. Okay, step 2, it kind of does this squat. The bacteriophage squats down, inserting its tail pins into the cell wall and breaking down the cell wall. And look what it can do next. It can directly inject viral DNA into the cell. Isn't that neat? So it squats down and then it injects its DNA into the cell, leaving the hollow shell outside. The capsid remains outside. So that means that the DNA got in and all the protein coat is outside. So in this case, do you see now why after penetration, uncoating is not necessary? Uncoating is only necessary if the capsid had gone in with the DNA, but the capsid stayed outside, only the DNA was injected, so uncoating was not necessary, right? And in this case, that DNA is free to take over the cell, and remember, now we're ready for synthesis, right? Duplication of phage components. That's called synthesis. Replication of viral genetic material. So remember, we want to make more of the genome, right? We want to make more DNA genomes of the virus, and we want to make all the little parts of the virus. So we want the capsid coat, the head piece, the helical tail, the icosahedral head, the tail fibers, the tail pins. We want the DNA genomes. Yeah, and then what do we want to do once we've synthesized all of our viral components? We want to assemble them into new virions. And look, here's the assembly process. We got to put all the capsid together with the DNA inside. We got to put the helical sheath here together, the helical tail with the tail fibers and tail pins. Yeah, there you go. It's a bacteriophage assembly line. And once we've matured, what happens? Do you guys remember? Are these enveloped viruses? No. So they do what? Look, they weaken the cell and lyse. You see? So the E. coli breaks open, it lyses open, and all these little virion pour right out, and they don't ever pick up a membrane. So these are naked, complex virion. And here's a nice little picture real quick. Actually, this is an actual picture with electron microscopy of the E. coli cell, this rod-shaped cell, breaking open, releasing all these little T4 phage, right? These little bacteriophage. Isn't that neat? And this is what the process of penetration looks like. Remember what I told you? The protein part of the virus stays outside, and only the viral DNA is injected into the cell. This is why uncoating is not necessary for bacteriophage. Isn't that neat? But do you remember that I also told you that a lot of these viruses, if they're termed temperate viruses, they can also enter a reversible state of lysogeny, which means that its DNA is incorporated into the host's genetic material. So what does that mean? Because what I just taught you what we just discussed was this, right? You see this right here? We just discussed the lytic cycle of the virus. That means the virus gets in, it constructs more of itself, it assembles, it ruptures the cell and gets right out of there to infect the next cell. That's called the lytic cycle. However, some temperate phage, bacteriophage, can enter what's known as the lysogenic state. So what does that mean? Let me show you. Watch this. Watch this. Now, if we don't want to go down that arrow on the right, we can go to the left. Look at this. The bacteria is infected with the virus. See, the DNA, the viral DNA is injected into the bacteria. And then look at this. Look what happens. Look at this. The bacterial DNA incorporates itself into the nucleoid, into the bacterial DNA. The viral DNA incorporates itself into the bacterial DNA. And look at that. So now you have bacterial DNA with the viral DNA embedded inside. It's like inserted inside. The viral DNA becomes latent as a prophage, and this is called a prophage. So when I say prophage, I'm talking about viral DNA that has inserted itself into the genome of the bacteria, and that's called the lysogenic state. And this is great for the virus because all the virus cares about is that its DNA lives on, right? So every time this cell divides, it's going to copy the viral DNA. Isn't that neat? So the virus doesn't have to do anything to propagate itself. Every time the bacteria divides, it's going to copy the viral DNA. And so it's an amazing way of just laying low. Why should you go through all the motions of replicating yourself and finding a new host if you can just lay low and have the host make many copies of you? Isn't that neat? So it's like a fun hack. It's like a cheat sheet hack for this virus to make many copies of itself, right? Now, what happens is this is all fine and dandy as long as this E. coli is having a good time and in a great environment, growing and very healthy. But the minute this E. coli runs into stress, right? The minute the E. coli runs out of, let's say, nutrients, it runs out of food, that's when the virus gets a cue to abandon ship, just like rats scurrying off a sinking ship. These viruses know, they have ways of sensing stress, especially the stress of lack of nutrition. So what happens is at this point, the viral DNA pops out again, and the virus can enter the lytic state and replicate, assemble, and release. Isn't that neat? So lysogeny occurs when the virus, wants to copy itself with minimal effort. But once the cell becomes sick, the virus knows the party's over. It will excise its prophage and it will enter the lytic cycle again to get out of there. And by the way, that process I just told you about is called induction. Induction is when the virus realizes, this guy's sick, I need to get out of here, right? That's called induction. Induction is when the prophage excises itself from the host DNA, and then it enters the lytic cycle. All right, and this leads us to concept check number 5. Which of the following is a step found in animal virus multiplication, but not in bacterial replication or multiplication? And remember, we talked about uncoating, right? Because the bacteriophage doesn't need to uncoat because it injects its DNA into the host cell. So C would be the correct answer there. I hope you got that one right. Next, the medical importance of viruses. Viruses are the most common cause of acute infections, affecting several billion people per year. Think of the cold virus, the flu virus, COVID virus. There's so many different viruses that can affect people. Some viruses have very high mortality rates. Mortality means death rates, including, let's think about Ebola, right? Have you ever heard of Ebola virus? When there's an outbreak of that, it's an emergency to keep it contained because it has such a high mortality rate. Next, now we're done speaking about viruses. Let's talk about other small infectious agents or other non-viral infectious particles, starting with prions. Prions are different from viruses because they don't have a nucleocapsid. They are literally a misfolded protein, a misfolded protein a protein that you already have in your brain and your nervous tissue. Isn't that interesting? Prions are essentially misfolded brain proteins. They contain no nucleic acid whatsoever. They are extremely resistant to sterilization techniques. These cause transmissible spongiform encephalopathies, which are fatal neurodegenerative diseases where the brain becomes spongiform, meaning so much brain damage that pores and holes appear in the brain. During autopsy, you can see them. It's common in animals. In fact, in sheep and goat, it's called scrapie. In cows, it's called mad cow or bovine spongiform encephalopathy or BSE for short. In elk, it's called wasting disease, and there's even human versions called Creutzfeldt-Jakob syndrome or Creutzfeldt-Jakob disease as well. And again, the way these work is that normal brain prion proteins, which are found in all of our brains, become misfolded, and that causes the disease. In fact, let's talk about the proposed mechanism for this prion disorder here. Here is a nerve cell. You can tell it's a nerve cell because it has dendrites at the top. That's where the nerve receives information, and an axon at the bottom where it sends that signal off. And look at this. Everyone has normal prion proteins inside of their nerve cells. See, normal prion proteins are displayed in green. In this case, let's say you ingested meat from a cow that has mad cow, and that meat contained the prion protein that's misfolded. See, the pink ones are misfolded, the green ones are properly folded. Well, if these misfolded proteins get into your neurons, they will bind with the normal proteins you have in your neurons and cause them to misfold as well. So it's almost like dominoes. Once some of these misfolded proteins are present, they will find the normal proteins and cause those normal prion proteins to misfold as well. And unfortunately, once these are misfolded, they form aggregates. They bind tightly together to form these long chains. And when those chains build up, they form fibers within the cell, which interfere with the cell's function and destroy it. Now you're having neurons get destroyed inside of the brain. And you can see these aggregates with the microscopy. And that's what leads to that brain tissue loss, the spongiform appearance, and the death. All right, now let's talk about other non-cellular infectious agents. There are satellite viruses, which are not real viruses. These are dependent on other viruses for replication. So for instance, adeno-associated virus replicates only in cells infected with adenovirus, or delta agent, a naked strand of RNA expressed only in the presence of hepatitis B virus. This was also called hepatitis D in the past. So these are usually pieces of RNA that depend on other viruses for replication. There are also these short pieces of RNA called viroids, which have no protein coat. They are only RNA, and they've only been identified in plants. So they cause plant diseases. All right, that leads us to our final concept check. Let's see if we can figure this one out. Exposure to nucleases. Nucleases are enzymes that chop up nucleic acids, such as DNA and RNA. Exposure to nucleases that degrade DNA and RNA would damage all of the following except, let's see, animal viruses. Oh, it would affect animal viruses because they have nucleic acid genomes. Bacteriophage also have nucleic acid genomes. Prions do not have any kind of nucleic acid, so it's likely Satellite viruses are usually RNA, and viroids are usually RNA as well, or definitely RNA as well. So the answer is C, prions. Why? Because nucleases would only chop up DNA or RNA, and prions are just misfolded proteins. I hope that makes sense. And that leads us to the end of chapter 6. I hope it all made sense. Now you're ready for exam 1. Remember, exam 1 covers chapters 1, 4, 5, and 6. Next, I'm going to be putting together an exam review video for you for those 4 chapters so that we can really get down and pare it down to see what you need to know for the exam. So let's do that and get ready for exam 1.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.